I'm having that be your separate section that I'm kind of, you can probably tell I've like, why do we have to include that in here? So we're doing what you need to meet the requirements of it being in the class, but it's not going to come back to haunt you a whole lot, so I don't want to put the energy into that. When it gets closer to the final, I will try and create a review sheet for you guys in there. All right, so the catabolic pathways. So this part here, this section is kind of why we go through all of the chemistry up until this point, is to be able to go through these reactions in here. We have this chapter, and then we have one on photosynthesis, and then the chemistry component of this class backs way down. It does not get more complicated in chemistry. Then we kind of switch into genetics mode of things. So your catabolic pathways are going to give energy by oxidizing organic fuels. And so your catabolism and ATP, you've got potential energy that's released in exergonic reactions. There's a couple ways you can get it. One is fermentation. With fermentation, you partially degrade a sugar. You don't completely degrade it. And it occurs without the use of oxygen. With aerobic respiration, oxygen is going to be consumed as a reactant with the organic fuel. And the term cellular respiration technically includes both aerobic and anaerobic processes. It got the name respiration in there because of it being tied in with your breathing to get the oxygen in there. So a lot of times that term cellular respiration is used generally to refer to aerobic respiration. I will tend to refer to them as your anaerobic and aerobic respiration or metabolism. I don't usually use the term cellular respiration because I try and avoid the terms that have a gray area in there for confusion. But it's there so you can be aware that not everybody will treat it that way. So the main reactions that we have is we can take an organic compound, various organic compounds, combine it with oxygen, and what you're going to get out of it is carbon dioxide, water, and energy. So if we look at it a little more specifically with, say, glucose, you've got the C6H12O6 plus 6 oxygens, gives you 6 carbon dioxides, 6 water, and energy in the form of ATP and heat. So your delta G of that is negative 686 kilocalories per mole. And that is under standard conditions. And so this catabolic pathway is not going to directly perform the cellular work. Instead, it's going to give you ATP that you will use to do the work. So what gets used in these are what we call redox reactions. They're reduction and oxidation, or oxidation and reduction in there. That means you're transferring electrons in there. So you transfer one or more electrons, it is a redox reaction. And redox is the common short name used for it. Oxidation reduction or reduction oxidation are also used, but redox is the short way to say it. So oxidation involves the loss of electrons from one substance to another, where reduction involves the addition of electrons from one substance to another. So the reducing agent is going to be the electron donor, and the oxidizing agent is the electron acceptor. So when something is reduced, you've added electrons to it. So for these reactions to occur, you need to have both a donor and acceptor. You can't just put electrons off into nowhere in there. So they're almost always going to occur together. Not all redox reactions are going to have complete transfer of electrons. Sometimes they will have a little bit of sharing in there. What we look at, they're going to be transferred in there, and that's good enough for what you're doing with these types of reactions in here. They're going to be transferred. It's pretty black and white on that. So if you look at this one here, you've got the CH4 methane reacting with oxygen, becomes oxidized, and then you have the oxygen become reduced, and it's going to produce the carbon dioxide energy and water in there. So you have one part of the reaction being oxidized, one part being reduced in there. And I'm not going to ask you to identify which part is being oxidized and which part is being reduced in a reaction. Just you want to know that they are occurring together. When something is oxidized, you have to have something reduced in there to go with it. So when we look at it with cellular respiration as a redox process, you've got your oxidation portion occurring here and your reduction portion occurring down here, with the oxygen becoming carbon dioxide and water. And so as electrons move, they're going to lose potential energy along the way, and it's released, which is how you are going to have a negative delta G in there, because energy is released out of it. And the energy state of the electrons changes, as does the hydrogen with its electrons as it's transferred. So one of the substances that you're going to see that's used for these redox reactions in metabolism to help move electrons back and forth is this NAD+, and NADH should be the other one that goes along with it in the electron transport chain. In metabolism, rather than just taking the glucose and breaking it down in one step to get the carbon dioxide and water, instead what they do is they break it down in a series of steps so that you can harness the energy along the way rather than doing the big explosive step in there. And so in the key steps, the electrons are removed from the glucose, and it travels with a proton as hydrogen rather than being transferred directly to oxygen. So it's first going to get passed to this carrier, NAD+. So that's where you'll see the NAD+ and NADH in moving electrons in a hydrogen. When it gets there, you have a dehydrogenase enzyme that's going to remove a pair of hydrogens from the substrate and oxidize it. So NAD is the most versatile electron acceptor in respiration. That's going to be where you're going to move electrons to in there. Electrons lose very little potential energy when they're transferred from glucose to NAD+, and the NADH has the stored energy that you use to make ATP. So it's kind of, you have another intermediate before you make ATP, the NAD in here. So you break it down, move electrons to this NAD+, and in moving those electrons you end up with ATP at the end. It will come together and make more sense when we actually look at the very process of it. So part of where you're getting energy is taking hydrogen and combining it with oxygen to make water in there. And we want to do this slowly in there because doing this reaction all at once would be an explosive release. That's why we want to use the electron transport chain to break it down into steps so that it's not an explosive reaction in there. And the reason that works is each carrier that you pass those electrons to is slightly downhill in the delta G, so it's going to want to move down there rather than to the uphill neighbor in there. And because we're continually adding oxygen into it, it's going to drive the downhill pull of the chain. because you're throwing off the equilibrium in there, so it never reaches equilibrium by constantly adding oxygen that you breathe. So when we look at the stages of cellular respiration, the first stage is going to be glycolysis. This is going to be a 10-step cycle that occurs in your cytosol, and you're going to take 10 steps to split glucose in half. And what you end up with are 2 pyruvates. Your next main step is going to be the citric acid cycle. It's also called the Krebs cycle or the TCA cycle. Your book is going to refer to it as the citric acid cycle. All the other books that I use call it the Krebs cycle. So I will be using those two really interchangeably in there, because I've made it a habit to always call it the Krebs cycle for my other classes. So this is going to occur in the mitochondria of eukaryotes, in the cytosol of prokaryotes, and it will completely break down the glucose. So it takes the pyruvate and breaks it down the rest of the way to make carbon dioxide, which we get rid of that by exhaling. And as it's doing those steps, you move electrons through the cycle, transferring them from NAD plus to NADH. When we look at the electron transport chain, it's going to go through oxidative phosphorylation. So you're going to put phosphorus on something, and it's going to involve oxidizing it as you do there. So electrons will be passed from one molecule to another down this chain of carrier proteins. And then at the end, you allow the hydrogen and oxygen to come together to make water. And you take the energy that you get from that making water, that explosive reaction, and you use the energy to make ATP that you get to use for something else. So we have 2 main types of phosphorylation that's going to occur. Substrate level phosphorylation takes a small amount of ATP that's formed either in glycolysis or the citric acid cycle when an enzyme transfers the phosphate group from an organic substrate. With oxidative phosphorylation, it's going to use inorganic phosphate in there. So that's the difference, where you'll see substrate level phosphorylation that uses organic phosphate attached to an organic molecule. Oxidative phosphorylation, it's inorganic phosphate. And sometimes when they mean inorganic phosphate, they'll say P with a little I by it. So for each step that you go through and do this, you're going to get about 38 ATP. You have to put a couple in, so it's a net of 36 ATP with about 73 kilocalories per mole of free energy formed. So these pictures, these are the ones that I put print me next to them on canvas for you that are in here. Is there a no that I just heard? Adenosine triphosphate. Yeah. be okay if I move it up a little? So when we look at glycolysis, there is an energy investment phase and an energy payoff phase. So if you think of it as you're going to start the barbecue to cook something, or you're going to start a fire, you've got to put some energy into it. You don't just look at it and go, I want a fire. At least I hope you don't do that. You have to pull out some matches and some other things to get that fire going in there. That's what we're doing with that energy investment phase, is we got to put some energy into this to try and get this fire going in there. So you take your glucose and you're going to have to spend 2 ATP of energy to get things going. And when you pull off the phosphate, that's what this is representing here, is the phosphate pulling off, an ADP, because now it's adenosine diphosphate instead of triphosphate. So then it goes into an energy payoff phase. And here you're going to actually get 4 ATP back out of it. So we say you have a net gain of 2 ATP because you've had to replace what you actually had to invest to get it started. And you're going to have some of that redox reaction going on, where you take NAD+, you throw in some electrons and hydrogen ions to make 2 NADH and 2 hydrogens there. And what you end up with at the end is 2 pyruvates and 2 waters. So your net out of the deal from the glucose is you get 2 pyruvates and 2 waters. You get a net of 2 ATP and a net of 2 NADH in there. A lot of times in biology when we talk about it, the waters get left off in there. I'm not going to hold you accountable as to how many waters you get out of things in there. If you can remember these other parts of the cycle, that's going to be enough of a challenge. But just know, you will see this in every biology class you take. Those waters come back. So in your energy investment phase, this is going to break it down step by step. You've got your glucose here. You put in the ATP, and it gets phosphorylated here. So you have one of the phosphates come off, and it goes right here. And it makes glucose 6-phosphate. So what this name means is that there is a phosphate on carbon number 6 of glucose. When you're memorizing these, sometimes people will abbreviate them with the first letter, like call it G6P in there. When you've heard the names enough, the abbreviations work really well for remembering it. So that's the first step. And then it's going to rearrange itself a little bit, and the enzymes are here. It's a hexokinase that does it. I put the names in here more so that you have them for reference of the names of the enzymes. Remembering the names of the steps will be plenty in there. So the next step will be to rearrange it a little bit, and an isomerase, an isomer is a rearrangement of atoms, so an isomerase is going to rearrange things in there. So it's a phosphoglucoisomerase in there. It rearranges it a little bit, so now it's fructose 6-phosphate. And our fire is still not burning very well here. So we have to put in a little bit more ATP to get this going again. It's kind of like when you go to start your fire and you put in the first match and it burns out. Yeah, got to try again. So a kinase is going to phosphorylate something. And so with another ATP, you get another phosphate put on there. So we have fructose 1,6-bisphosphate. Bis means 2. You may also see it as di in there in the name of it. So it means you've got a fructose with a phosphate on carbon 1 and carbon 6. So now we have invested energy in there, enough energy that the thing is going to split in half. And when it splits in half, you get this dihydroxyacetone phosphate and this glyceraldehyde 3-phosphate. And what's going to happen is this is going to be in equilibrium, an equilibrium reaction where it can be reversible here. However, this is the one that's going to get used up. So what's going to happen is it's going to cause all of these to be converted to this. So sometimes when you see this, they'll actually leave out this step because this is all going to get converted to the glyceraldehyde 3-phosphate in there. And so the isomerase catalyzes the reversible reaction of the dihydroxyacetone to make the glyceraldehyde 3-phosphate. And I think, did I type these all out for you in the outline in there? Okay, good. So this is just a word reaction of what's going on in this picture. Unless you are a person that really needs to look at the word reactions, for most people, looking at this will probably be more helpful to visualize this process than reading all of those words. So with the energy payoff phase, we've got that glyceraldehyde 3-phosphate here that now we're going to use the triose phosphate dehydrogenase to catalyze the oxidation of it to 1,3-bisphosphoglycerate. So you're going to be doing some of that redox stuff here to make that NADH. Now we're finally going to get a little bit of energy back out of it here. So you're going to have those ADPs that you get from using energy that are going to come along, and they're going to pick up the phosphate on there. So you have another kinase in there that's going to phosphorylate ADP to make ATP. So this time you're going to be releasing the energy and putting it on there. So it's lost one of the phosphates. You actually have 2 of these for every glucose. So that's why they're showing 2 ATP in there. And now instead of it being a bisphospho something, it's just 3-phosphoglycerate because one of those has been pulled off. Then we rearrange it a little bit again to have 2-phosphoglycerate. And we rearrange it a little bit more again to have phosphoenolpyruvate. So still has the phosphate on it. And then we have another kinase that's going to pull off another phosphate off of each one. So remember, you've got 2 of them that go through the cycle. And you end up with pyruvate at the end. So if you are looking at those steps on there of metabolism for glycolysis, what I want you to be able to do before I move on is say, okay, I can follow those steps and see where the phosphates go in there and what happens to the carbons. So you start out with all of your carbons at the beginning. You haven't gotten rid of any carbons here. You still have 6 at the end. You can follow it on there and look and see that's where the phosphate goes. This is when it comes off on there. So everybody can see on those diagrams of the reactions where that happens. Okay. The other part of memorizing is just one of those things that you pound it in there. So we've got this pyruvate here, and all we've gotten is 2 ATP out of the cycle at that point. 2 ATP is really not very much. 2 ATP doesn't allow you to do a whole lot. So we need to continue breaking it down, and that's where we will use the citric acid cycle or the Krebs cycle here. And in eukaryotes, this is going to go into the mitochondria. If you're a prokaryote, you've got part of your cell membrane that folds in that you use to do this in the cytosol. And you're going to have oxygen present for this part of the cycle. So what happens is you've got your pyruvate here that you've made. And what you're going to do is you're going to pull off carbon dioxide in there. But you need to have oxygen present in order to do that. If oxygen is not present, this won't come off. And when you're moving oxygens like that and electrons, we get more of this NADH thing again. So this is a way of temporarily storing the energy because you've moved those oxygens. And what you end up with is acetyl-CoA. It's acetyl because you've got 2 carbons left on the sugar. CoA because you have coenzyme A joining onto it. So coenzyme A is derived from B vitamins. So this is one of those places where B vitamins are important. If you're lacking B vitamins, you can't do this step. So it's the cofactor in there. Most of your B vitamins, you're going to get those from fresh fruits and vegetables pretty easily in there. So it does this, it makes this acetyl group very reactive for when it's fed into the Krebs cycle. And being that you have these words here, I am just going to skip down to the picture to show it. So you've got this acetyl-CoA that comes in. It's adding 2 carbons to the mix. It's joining with oxaloacetate that you have left over from the previous turn of the cycle. Oxaloacetate has 4 carbons in it. 1, 2, 3, 4. So what it creates is citrate that they name the cycle after that has 6 carbons in it. So the citrate, again, is going to get rearranged to make isocitrate in there. Still has 6 carbons. And then it's going to pull off one of the carbons. And it has to have oxygen to do that. That carbon attaches to an oxygen molecule. So that's going to be more carbon dioxide that you're going to end up exhaling in there. Now you're down to 5 carbons. You've moved electrons again, so a little more energy is released as NADH. And you have the 5-carbon alpha-ketoglutarate. So you're going to pull off another one of the carbons in here. And when you do that, more electrons move, so you get more NADH. And then the coenzyme A hops on here again. And you end up with succinyl-CoA that only has 4 carbons in it. And then we're going to rearrange this to succinate. We pull off the CoA portion. And we're going to get more energy released. This time it's going to be released in the form of GTP. And then that GTP is very similar it ends up transferring its phosphorus onto an ADP to make ATP. So then we're going to take that succinate and rearrange it again. So every time we rearrange this, we're taking it to a slightly lower energy state in there. So it's still having that negative delta G, and it's just picking off a little bit of energy for that delta G at a time. And it's going to get a little bit of energy released as FADH2 this time. So if it helps you to think of these things as all being a type of energy currency, your ATP is like your dollars, your NADH would be some euros, FADH2 some pesos in there. They're all energy currency, but at the end you're going to convert into dollars. So we rearrange the fumarate a little bit to make malate. Still has 4 carbons. And then we rearrange that malate back to be oxaloacetate again. Moving some electrons, we get more NADH off of it. And we're able to repeat the cycle. So from 1 glucose, you've made 2 pyruvates that end up as 2 acetyl-CoAs that go through that cycle 2 times. So, able to look at your picture and follow where your phosphates and carbons go? I can tell you when all of this stuff finally made sense to me, I went and celebrated with a nice run and a big pizza. So right now what we've ended up with is a whole bunch of these NADHs in there and some FADH2, and the cell likes to burn ATP. It's kind of like if you went over to Europe and you stopped in Canada on your way back and you had all this foreign money and you go to the grocery store here. They're going to say, it's money but not the kind we're taking. That's the same thing your cell is going to say if you try and do work and spend NADH to do it. It isn't interested. It says go exchange it. So that's what we do in the next step in here is we use chemiosmosis to do this in oxidative phosphorylation. And it's your electron transport chain. It's in your mitochondria. It's a collection of molecules that are embedded in the membrane that are literally going to pass electrons down this chain in there. So you've got these multi-protein complexes, and they all get names, 1 through 4. And then they're going to be tightly bound to these non-protein prosthetic groups on there. And these carriers, as they pass the electrons, they go in between being oxidized and reduced back and forth as these move down the chain. So you've got your NADH here, and it's coming up to its little money exchanger here. So it's going to take off its electrons, and it drops them off with FMN. And it's going to pass the electrons along the way here. You've got this iron sulfide in here, and coenzyme Q that it's going to pass it to. So one of the reasons why you need to have oxygen or iron intake in your body, and a person will feel fatigue if they don't have enough iron, is it's in this electron transport chain. It's part of what carries your oxygen on hemoglobin, but it also carries electrons in this electron transport chain. So if you don't have that, you're not able to exchange everything to get all of your ATP back in there. FADH2 is going to pass its electrons off it cuts in line a little bit in here and passes them off. It skips over FMN and it just takes it to the iron sulfide in there. And those electrons come in at your coenzyme Q here. Then they move on to the third complex here, where it passes it along. These electrons are getting passed along here in between site B, more iron sulfide, site C. And the sites are cytochromes in there. Until they get to complex 4 here, where you have cytochrome A3, and it's going to pass these electrons out the other side of the membrane. So what you've done is you've passed these electrons along this membrane in here, and they get to the other side. So now you have protons and electrons that are separated on 2 different sides of a membrane. So what happens when you have positive and negative separated? They want to come together. It's kind of like when we take over there and we have the boys and girls separated. They want to come together. And if you've seen them in the cars, you know what they do. So what will happen is we're going to allow these to come together with oxygen, and it will make water, and you're going to get energy out of the deal. So at the end of the line, we've got this enzyme, this ATP synthase enzyme that's going to be in the inner membrane of the mitochondria, and that's going to be where we allow those electrons to come together in exchange for phosphorylating something. So we can say we do this by chemiosmosis because you've got an osmotic gradient in there formed by these chemicals separating the hydrogens, the positives, and the electrons, the negatives, in there. And so that ATP synthase works on a rotary motor in there. And I think, did I type out this stuff for you too on the ATP synthase in there? So what's going to happen is you've got this rotary motor in here where you're going to have the hydrogen ions come in and the thing spins around. When it gets almost all the way around, it's going to drop it through the next tube in here. You've got the catalytic knob, and then when they come down, those electrons get to go be with their hydrogen ions and make water, and it has fueled this process in here, spun this around to put the phosphate back on the ADP to make ATP. I didn't realize we have actually gone over the time in there. So we will stop there today with things. Like I said, if you want to come back at 11 o'clock, I can kind of go through and put a summary together for you of this stuff with glycolysis of what we've gone through in there. We'll still have the test next Friday, but what I am going to do is I will put together some additional stuff for you over the weekend too, kind of helping to try and tie this together for you in there. We'll have a little bit of time on Wednesday when you've got downtime where you're just watching things. One of the things you're doing in the lab has to sit for 30 minutes in there, so we'll have a little bit of downtime in there on Wednesday that we can go through this stuff a little bit more too.